Hi, and welcome to episode 76 of the Breaking Bio podcast. I'm Morgan Jackson, a PhD student at the University of Guelph in Ontario, Canada.
I'm Heidi Parker, a postdoc at the University of Texas at Austin.
Hi, I'm Tom Housley, and I work at the University of Cambridge in the UK.
And this week we're joined by Dr. Yoel Stuart, who's a postdoctoral researcher at the University of Texas at Austin. Hi, Yoel. Thanks for joining us this week.
Hey, thanks for having me.
So what do you do at Austin as a postdoc?
Well, I'm currently in Dan Bolnick's lab, where we study the evolution and ecology and immunology of stickleback fish. So we have a lot of different projects that we work on. The one that I'm working on in particular is studying sort of using the stickleback to study how deterministic evolution is. So if we give organisms the same set of starting conditions, will they end up in the same place, or will they end up in a different place? And if they do end up in a different place, can we ask why and how? So we've got a bunch of replicated populations of stickleback that are independently invaded a bunch of lakes and streams up in Vancouver Island, Canada, and so we're using that system to ask those questions.
So if we can back up a little bit, so you had a science paper several weeks ago, but that wasn't about fish, it was about lizards. Do you want to tell us kind of the brief synopsis and punchline of this?
Sure. So that was a study of Anoles lizards. Anoles, you can find them in the southeast U.S., but also there's many, many more species that live in the Caribbean, all the islands in the Caribbean, as well as Central and South America. And what we were interested in was how the invasion of one Anoles species into Florida, the invaders of the brown anole, Anoles sagrii, may have caused a change in both the ecology and then driven evolution of a native species in the U.S., Anoles carolinensis, or the green anole, which lives in Florida as well as other states in the southeast. So what we did is we went to a bunch of small islands off the Atlantic coast of Florida. They're actually in a little lagoon. They're in a lagoon system, and these islands are man-made. They were created in the 1950s when the Army Corps of Engineers dredged a shipping channel, the Intracoastal Waterway, through the lagoon system. And when they dredged the channel, they basically created a bunch of small islands with the dredging material. And they created them, they're all about the same size, they're all about the same distance from the mainland, and they're all the same age, and they've all sort of subsequently developed this really nice, forest system. So they have a bunch of trees, and all the mangroves grew around them, and they have this, full set of, or almost full complement of plants and animals on these islands. And so basically they serve as these nice little natural experimental laboratories. And the cool thing for us, interested in this interaction between the two species of lizards, was that on all these islands we found the native lizard, but then on some of the islands by the late 80s, early 90s, the invasive lizard was starting to get to. And we had good expectation that these two species should compete strongly. They're both about the same size, they're both active during the day, they both eat insects for prey, they both hold territories. And so what we wanted to know was, well, if you've got this invasive coming in, competing with the native, what's going to happen to the native? How will it respond? Will it go extinct? Will it keep the invasive from coming in? Or will it change in some other way to sort of adapt to that native? So we want to know how competition between those two species would play out. So in the mid-1990s, my colleague, whose name is Todd Campbell, he's a professor at the University of Tampa, but at the time he was doing his dissertation at University of Tennessee, Knoxville. He wanted to know, what sort of the demographic effects of the invader would be on the native. Would the populations crash, or would they stay at the same level? What would they do? So he introduced the brown anole, the invader, onto 3 islands, studied the green anole on those 3 islands, as well as 3 other islands as controls. And he saw a couple of things. First, he saw that the green anole populations crashed. They went, from being pretty common to much less common. And then sort of more interesting or not more interesting, but then more relevant to the rest of the story, he also saw that the green anole, the native, also moved higher up into the trees. And then on the biggest islands, it didn't, even though the populations were reduced, didn't go extinct. So it sort of seems like this move up into the trees that he found is the way that the green anole was able to sort of get out of the way of the brown anole, move into a new part of the habitat, and thereby coexist with this invader that came in. And from there, what I wanted to do when I came back for my dissertation about 15 years later was ask, well, we see this ecological habitat shift here. You've got this lizard this native that's adapting to an invasive by moving up into the trees. Perhaps that imparts or imposes some sort of natural selection on these natives to adapt and evolve to this new part of their habitat. So basically what we wanted to know was, is there this rapid evolutionary response by the native in response to this habitat shift that it made? And so I went back in the 90s, I went back 15 years later, so it was about 2009, 2010 by then, and I wanted to go back to those same islands, the same experimental islands that Todd Campbell had worked on, and ask if the lizards had evolved, had they adapted in some way. And it turns out that I couldn't use his islands because all of his control islands had been invaded subsequent to the end of his dissertation. So I sort of had to regroup a little bit, and I just basically went and I found 5 uninvaded islands. So 5 islands with just the native anole on them. And then I picked 6 other islands that had both the native and invasive. So I kind of had, rather than this like direct experiment, I had this nice natural experiment where I could say, all right, what's the green anole, the native anole, doing on those invaded islands and compare it to the 5 uninvaded islands. And the prediction, well, we had 2 questions. So we wanted to make sure that perch height shift, that move up into the trees, still existed. And so we went onto the islands and spent, we'd sort of boat out. These are small islands, they're about a football field in size, and they're probably about 50 to 100 yards from one another. So we had a small boat that we'd boat out to all the islands. We'd land in the morning. We'd spend most of the morning into the early afternoon just kind of walking around slowly until we saw a lizard that we hadn't disturbed. We'd measured its perch height, et cetera, for all 11 islands that we worked on, just to make sure that perch height shift was still there, and it was. So then we felt pretty comfortable having this basis to expect an evolutionary response. And the evolutionary response that we expected, that we predicted, was that the lizards that moved up into the trees would evolve these larger toe pads. So like geckos, anoles can cling to surfaces, they can climb up walls, and they can climb on leaves and things. And the way that they do that is with specialized scales on their toes, on their toe pads. These scales have thousands and thousands of little hairs on them that let them cling to surfaces using van der Waals forces, if you remember your high school chemistry and such, and physics. And we know also that across the 400 or so species of anoles, those anoles that tend to live higher up in the trees also tend to have larger toe pads. And we also know that larger toe pads relative to your size gives you a better ability to cling. So it seems like there's this sort of adaptive, this adaptive role of toe pads, whereas if you're higher up, you're better at clinging, you're going to have larger toe pads. And so probably to sort of make you better at climbing and moving and interacting with other lizards while you're doing your lizardy things up in the trees. And it probably lessens the chance that you'll fall out of the tree and then be subject to predation and things. So we had predicted that the toe pads of the native anole on the invaded islands would get larger once they moved up into the trees. So while we were taking those perch height measurements, we were also collecting the lizards whenever we could catch them. We would use these little extendable fishing poles that you can just that had a little lasso on the end of the pole, so you get the lasso around the head of the lizard. You give it a little tug, and it comes with. The lizards are fine. They're so light that it doesn't hurt them or anything like that. We brought those lizards that we captured back to the lab. anesthetized them, stuck their toes on a digital flatbed scanner, like a document scanner, like something you might pick up at Staples. Took an image of their toes, like a little fingerprint, and then the lizards would wake up, and we put them back where we found them the next day. And so from those images of their toes, we were able to count up the number of scales as well as the area of the toes to see if on invaded islands the native anole had gotten those larger toe pads like we predicted. And once we analyzed the data, we found that indeed they did. So it seems like they have larger toe pads on these islands. They're probably better at clinging, and they're adapting rapidly. Within 15 years, which is about 20 Anoles generations, we've got this nice shift in these toe pads in response to the arrival of this invader.
Were you surprised how quickly that happened? Because 15 generations is not long.
Yeah, I think that one cool thing in evolutionary biology that we're, especially us field ecologists, field biologists, starting to realize, but I think that the sort of fruit fly and microbial folks have known for a long time, is that if natural selection is strong enough, evolution can happen quite rapidly. The Darwin's finches and the Trinidadian guppies are good sort of examples of rapid evolution when you have this strong selection. So I think that we had predicted that evolution would happen, but still in your gut you're always kind of surprised by when you actually do go and find it, because like you're sort of implying, the general dogma is that evolution is always so slow and it takes hundreds and thousands of generations to see it, and so even though we predicted that we would see it, we were still surprised that we found it, against that kind of gut feeling, I guess.
If it had gone longer, would it have extended your PhD longer?
Yeah, probably.
It's like, his PhD was 32 years. We get a science paper.
Right. I mean, in some sense, though, the study was 15, 17 years long. My colleague Todd Campbell started it in the mid-90s. So we know that the invasive got to the islands that I just talked about the evolution component of the study. We know that the invader got there sometime between Todd's study and now, and so his study basically let us, because he surveyed all of those islands for both species of lizards, and so his survey sort of lets us bound down the earliest time that the brown anole could have gotten to the islands that we worked on. And so in some sense, the experiment did start 15 or 17 years ago. So, and spanned the course of 2 PhDs plus more time. So, yeah, it's funny that we want these. I was at a talk once where the woman was giving her talk, and she was up there and she was like, yeah, we only have 30 years of data on this system. And my friend who was next to me kind of perked up at that. He was like, 30 years, that's a huge amount of time of data in a field system. So we'd really love to have all these long-term data sets, but they're just so hard to get, and because we have such short granting periods. And so we were lucky in this case that we were able to sort of peg together two different projects and have a longer-term data set.
So you obviously had really cool morphological change over that period, but did you find any behavioral responses apart from, you said that they're higher in the trees, but were they going for different resources so as not to be competing with the invaders as well?
Yeah. So I had a student who was working with me who was looking a little bit at sort of whether their behavior changes in how much they move. So do their movement rates change, once they move up into the trees and once they're interacting with the invader? And she's actually working on this project. I think she submitted the manuscript from this yesterday, actually. But she did find some changes in movement rates, seemingly affecting males a little bit more than females. So females kind of move at about the same rate, but males move less on invaded islands than on invaded islands. Male green anole. And then as far as the diet stuff goes, that's sort of the next direction that we want to go. We've seen this habitat shift, we've seen this evolution, and we think we've done a lot of work to basically show that it is this interaction between the two species that's driving this habitat shift. And the next question that we'd like to get at is, well, what is the mechanism of that interaction? Is it that they're competing for food, like you suggest? And so the green anole is moving up in the tree to get different food resources, or more available food resources, or maybe it's just that they interact really aggressively with one another. So the green anole, just moves up in the trees to get out of the way of this aggressive brown anole. Or maybe to sort of take that to the extreme, we do have some evidence that the brown anole will eat green anole hatchlings. So it may just be that those green anoles that move up into the trees right away and have the ability to do that with their toe pads are the ones that are selected for. And probably it's a little bit of all of those things, And so the future, and those are quite hard to tease apart, and so that's where the future directions lie, for sure.
Do you ever have hybrids of the brown and the green? Do the females ever mate with the wrong?
There's reports here and there of cross-species copulation, but we haven't ever seen any reports of actual hybrids formed. They're about 20 million years diverged. They have very differently colored dewlaps, or these flaps of skin under their throat that they use to communicate. And so that may occasionally happen, but I don't think it happens a lot, if at all, on some of these.
Yeah, it's such a cool system. It seems like there's so many directions you can take it. I hear anole people sometimes saying, "Oh, it's so flooded. The field is so saturated," which they should probably look at some model organisms and really see what saturated is. But how did you just go ahead?
Sorry, I was going to disagree. I don't think it is. I think that there's a lot of anole studies, but it's one of those balls of yarn kind of things. The more you know, the more you know what you don't know. And the more you realize that, there's a huge paucity in behavioral work on Anoles. There was a little bit of work done on behavior in the 70s and 80s, and then a huge gap until, now, essentially. I know a couple of folks who are working on, Anoles behavior now, but there's a huge amount we don't know about how they interact with one another and what they eat and why and all these kinds of things. So it's not flooded. Everybody should study Anoles.
Except now you're working in fish. So how did you decide to change over? So the generation time?
Well, the stickleback are definitely easier to keep and grow in the lab, but I'm not actually doing any live animal work right now. The stickleback are, as a evolutionary ecologist who's interested in how these differences in ecology drive evolutionary pattern and evolutionary chain. Stickleback are really powerful in that regard, in that we do know a huge amount about their ecology, what they eat, where they live, what other species they interact with, and how differences in their ecology drive differences in their morphology and evolution and things. So the stickleback are a really powerful system for asking some of these questions about ecology and evolution and their interplay. And stickleback are also quite nice in that they have a huge amount of genomic tools, so we can then also get at the genetic components of those evolutionary ecological questions much more easily. So in this case, switched to stickleback because I was really interested in this question about how deterministic is evolution, and how do ecological differences influence evolutionary patterns. And these stickleback populations that we're working with are highly replicated, so it's this nice natural experiment where basically nature has started the experiment for us, and now we can come in this case, not 15 years later, but about 10,000 years later, and say, well, what's happened subsequently.
So you guys are working with comparing sticklebacks, which are freshwater fish, right?
Yeah.
and you're looking at how different pond and stream communities that you I assume think may have been previously, the same population have diverged, and how that's happened multiple times, and how their behavior and their morphology may have changed with each of these new incidences into a different stream or habitat?
Yeah. Yeah, so stickleback, I should correct myself a little bit. Stickleback both have freshwater and marine populations. And it seems like the marine population is kind of this big homogeneous population that then seeds all of these different freshwater invasions. So we kind of have this putative ancestor that we can go back and compare to, and then say what happens when stickleback go from saltwater to freshwater. What sort of adaptations do they make? And there's been a lot of work done on that saltwater freshwater transition. So the stickleback will tend to lose a lot of their armor plating and a lot of their spines, and the genetic basis of those changes have been looked at a lot. But we're sort of more interested in the lake to stream transition. So as stickleback populations are adapting to lakes versus streams, how are they doing that, and why? And so some of the sort of general patterns that come out is that lake fish tend to be much more streamlined because they do a lot more swimming, and they tend to eat a lot more plankton prey, and so their jaws and faces tend to match, and trophic traits tend to match eating the plankton prey, whereas stream fish tend to be a lot bigger, a lot more deep-bodied, and their trophic characteristics match the fact that they eat a lot more prey. Macroinvertebrate prey that's down on the bottom and on the benthic surfaces of the streams, and so that sort of general pattern has been known for a long time. But what we've done is we've gone to 16 independent watersheds and picked 16 independent lake stream pairs within those watersheds, and then basically, in whatever space you like, in morphological space, if you have size on one axis and mass on another axis, and then you can sort of imagine a z-axis and then a whole bunch more axes. We can plot the sort of mean of the lake fish and the mean of the stream fish, and then we can draw a line between the two means, and that's sort of the direction of divergence that the lake and the stream populations have traveled along. And then what we're doing now is we're basically saying, all right, if that's the direction of divergence for this watershed, what about this watershed? Is it the same direction, which would be a parallel evolution, or is it a different direction, which would be non-parallel evolution? And so we can sort of quantify the degree to which evolution proceeds in the same way across all these different watersheds.
So how do you? I mean, we're going to go to the very basic chicken before the egg here. How do you decide or figure out which came first, the stream or the lake population, and does that impact how you're figuring out these divergences and convergences?
I mean, the fish would swim, most likely just swam upriver, so they'd be in the streams before they got to the lakes in this case. But I think we're sort of assuming that it need not necessarily matter which one came first. They'll both be about the same age, and they'll both have been evolving for about the same time. So we're just kind of saying, all right, it may not be fair to assume that they've reached equilibrium, but let's say that they're, on their way to that. And we're just sort of saying, all right, now, 10,000 years after, these lakes formed on Vancouver Island, what's the divergence look like between lakes and streams? And, that you're sort of hinting at the fact that there may actually be, a huge amount of variation in what sort of the starting conditions are for some of these lake stream pairs. Like not a lake stream pair isn't a lake stream pair isn't a lake stream pair, because maybe some lakes are younger or older than others, or because maybe there's more gene flow from other populations of the marine environment in some lake stream pairs versus another, or maybe just because one lake is definitely not like another. We collected from one lake where you can walk across it, and another lake where it's tens of meters deep in the middle and a mile wide, and then we had streams that were fast flowing and streams that you couldn't, it just looked like a puddle, and so that's kind of, that variation is actually something that we're quite excited about, because we're going to try and use that ecological and genetic variation in these populations to explain this morphological parallelism or non-parallelism. You know, like we've got 16 pairs, 4 or 5 or 6 of those pairs are diverging in one way, 4 or 5, so they're all in parallel, but then there's another set of like 4 or 5 or 6 that are kind of diverging in parallel another way, and then we've got some weirdos that are just, you know, going all around in circles. And so we're excited to try and explain all of those differences with the ecological and genetic variation that we've measured.
So you kind of just touched on what I was going to ask there, which is how do you go about deciding what you're going to use as your kind of predictor variables for your responses? Because you can go and measure all these morphological traits, but like you said earlier, you know, there's some which, if they're in the lake, then they do more swimming, so a behavioral trait will influence morphological traits as well.
Yeah, it's a good question. If you have any ideas, let me know.
Collect a lot of data.
Yeah, I mean, essentially that's you know, we've done that in a sense. Is all right, we'll just think of everything, you know, we can to collect. And now I'm sort of sitting on all that data, and I'm trying to decide best way to go after it. And you know, sticklebacks have been studied enough that there are a number of sets of sort of morphological characters. You know, you've got your trophic traits, your swimming traits, your full body traits, everything thrown together. So we'll probably go at the morphology in those kinds of units, and then you know, we're still trying to figure out how to bring in the rest of the multivariate data set into. I mean, we can look at this, you know, these same axes that I drew for parallel, non-parallel morphological divergence. We can do that for genetics and for environment as well. So we can say that you know, these lake stream pairs have parallel environmental divergence, and then we can try and correlate that parallelism with parallelism in morphology to say, well, there's a correlation between, you know, between the environment and the morphology, and we can do the same for the genetics. But it's a huge data set, and we're still trying to figure that out.
So do you have differences in the mating systems across different populations and species and things with the sticklebacks?
Yeah, so other folks in the lab are doing a little bit of that work, but there's definitely differences in environment among lakes and streams. You know, some have really clear water, some have reddish tinted water, some have bluish tinted water, and it seems like, and then there's also differences just by depth, and we've got, you know, some data, and it's not mine, so I'm just sort of reporting secondhand, but got some data suggesting that, you know, in different populations they'll have different, you know, ability to detect differently colored light, and they'll use those different light cues to choose mates. They'll also use depth cues to choose mates. Females, I think, if I'm remembering right, some of the results that we got is that females prefer males that live in shallow environments, or as Dan likes to say, females prefer shallow males. But then they have, I think that males living, well, I shouldn't speculate because I can't quite remember exactly what it is, but yeah, there are differences in mating systems and techniques among ponds, but I don't have too much detail for you there. Sorry.
Yeah, no. I was just wondering whether this is going to create another whole axis of terror for your analysis.
Well, we're not there yet. I mean, I was just going to say another axis that we're hoping to get to in the future is we've measured ecology, genetics, and morphology, and we'd like to actually try and measure parallelism in natural selection in the wild. And so, you know, we've got hopes and dreams to do some crosses and introduction experiments with F2 lake stream hybrids to basically say, you know, what are they? You know, we assume that parallel environment means parallel selection, but we'd actually like to measure selection and see if it's, you know, asking for the same things out of these populations every time. So that's another direction we'd like to go as well, and then we'll go to the mating system route, and then we'll, you know, just keep going. So.
I was just going to say, it does seem like you said with the Anoles, like it's this ball of yarn where you can get deeper and deeper at these questions.
Yeah.
Really such a It is. It's so important picking your organism.
Yeah,
You know, that
absolutely.
you want something that has some genetics and yeah.
Yeah.
Well, it certainly sounds like you got plenty of work ahead of you, slaving through some rather excessive data. Where can people follow along with the successes and possible angst that you're facing in the next few weeks or years?
I don't know. We don't have a Bolnick Lab blog, but maybe we should start one of those. Although every now and again, some of our stuff pops up on Andrew Hendry's evo.blogspot.com blog, and then of course any of the Anoles stuff you can go to anoles.org and follow it there. But otherwise, keep reading the literature; it'll be out there eventually.
Awesome.
Like three or 4 years from now, you know.
With any luck, that'll be what it is. And you're on Twitter as well, correct?
I am. Although I'm not as good at tweeting as I maybe should be. But yeah, I will tweet out some things now and again. So I think it's just at Yoel Stuart is my handle. Is that what it is?
Yes.
That's what we have on record. So.
All right. Sounds good.
Awesome. So we will, like always, make sure that we put links to all these various places, as well as some of the papers that we talked about today with Yoel, as well as his Twitter handle, so he can check it, and anybody listening at home can check it as well. We'll post all of those on our website at breakingbio.com. We got to wrap it up now, but we need to thank the European Society for Evolutionary Biology for supporting us with some outreach funding. And if you're listening at home, we'd like to hear from you. Drop us a line on Twitter by following us at Breaking Bio. You can drop us a comment at Facebook by searching for the Breaking Bio podcast. And, of course, you can take us with you by searching for us on iTunes for the Breaking Bio podcast as well. And we'd love it if you'd leave us a rating or a comment or something along those lines there. Just lets us grow our audience a little further and help bring some of this cool science that we listen to and talk about to a broader spectrum of people. That's it for this week. I'd like to thank Yoel for joining us, and I'd like to thank everybody at home for listening as always. Talk to you next week.